Welcome to SUNY Poly's Graduate Student Research Showcase. I'm Krista Thompson, Assistant Dean of Graduate Studies. We're delighted to have you here with us. Today's presentations will be provided by PhD students in nanoscale science, nanoscale engineering, nanobioscience, and from the MD-PhD program. At the conclusion of the presentations, new students will have the opportunity to ask questions. Without further delay, I'd like to introduce our first presenter, Pujitha Ramesh.
Of fibrosis and regeneration of the salivary gland. So when you think of fibrosis, it's a chronic condition that can happen in any part of the body where there's been an injury and wound healing has begun, but in the process goes wrong, and instead of having just regular extracellular matrix being deposited, there is excess extracellular matrix being deposited. So cells in the human body are not free floating. They're in this matrix which provides both biochemical and mechanical cues for the cells. And this matrix is called extracellular matrix. When there is a wound, usually there is extracellular matrix deposition so that wound can heal and cells can migrate into that region, and the gland can regain part of its function. In fibrosis, what happens is that excess extracellular matrix is deposited, and so the cells are not able to migrate into that region. It forms a scar tissue sort of, and the biochemical and mechanical cues are all wrong, and the cells are not able to regain their function. They start behaving in all sorts of odd ways. And the same thing happens when there is fibrosis in the salivary gland as well. There isn't enough saliva being produced, and the current therapeutics that we have, which are pilocarpine and cevimeline, let me show the pointer here. Yeah. So these are not very effective in improving saliva production. And most importantly, the side effects from these therapeutics are far worse than the symptoms of the condition itself. So we here propose an alternative therapeutic strategy wherein we would use cell therapy and regenerative therapy to regenerate the function of the gland. So mesenchymal stem cells have proven to both reduce inflammation and fibrosis in different parts of, say, fibrotic glands all over the body. And the issue with directly injecting mesenchymal stem cells is that most of them do not engraft into your target site. Whereas if you kind of encapsulate them in a scaffold and then implant the scaffold cell construct into your injury site, then there is a much better chance of these MSCs engrafting onto the gland. And also, if you fabricate the scaffold in such a way that the native cells are able to migrate into the scaffold, then the gland can regenerate a portion of itself and regain function. So this is the therapeutic strategy we've proposed. And to fabricate that scaffold, you need a scaffold which has enough interconnected pores, with the pore sizes being in the range of about tens of microns, because cells are of the order of 10 to 20 microns. You would want the matrix to have both an insoluble backbone and a soluble cushion, so that the cells can kind of lean on the insoluble backbone, but they also have this cushion that's supporting them. They should also have enough cell attachment sites, because if the cells do not attach to the matrix, then they would die. And it should have the right stiffness. It should be of the order of about 100 to 300 pascals. So when you look at the current state-of-the-art scaffolds available, all of them don't tick all of the boxes at the same time. So we set about to fabricating a scaffold that would have all of these properties, and we use this emerging technique called cryogenic electrospinning. With cryogenic electrospinning, you can make a nanofibrous mesh in 3D, but we modified the technique to fabricate a mesh which had this honeycomb topography as shown here. And we were able to fabricate it to have properties topographically similar to the native decellularized adult salivary gland, ECM. And we were also able to achieve similar stiffness in the order of about 120 pascals and similar relaxation half times. We were able to incorporate soluble and insoluble components of the ECM backbone. and to test if the pores are able to allow cells to penetrate and if the cells are able to stay alive and grow. We seeded cells in them. A vast majority of the cells that attached to the scaffold remained alive. They were able to form deep three-dimensional clusters. And apart from the cells being able to thrive, they should also be able to retain and maintain their function. So we tested for specific proteins being expressed by these cells, as shown here in the right here. And the cells did express the proteins where they should in the cell, and they were able to maintain their function. So because this was a collaborative effort, there were a lot of people involved in it, and I'd like to thank all of them. And thank you for your attention.
How's it going, everybody? My name's Zach Olmsted, and I'm an MD PhD candidate in the lab of Dr. Pellew. So I actually went to SUNY Poly as an undergrad and graduated with a BS in nanoscale engineering. And I'm currently in the 5th year of my dual nanomedicine program and in the 3rd year of my PhD work. So this talk is entitled Transplantable Neurocircuitry for Spinal Cord Injury. And it's funded by the New York State Spinal Cord Injury Research Board, as well as SUNY Poly internal seed funds. So what is spinal cord injury? This is an abrupt and traumatic injury directly to the central nervous system in the spinal canal. And so if you sever the axons that connect the peripheral body with the brain, you disrupt the conduction and the information flow between the brain's higher centers and the rest of the body. And since the nervous system wires all other organ systems in the body, this will have a direct or an indirect effect on all organ systems. And because of the non-regenerative nature of the central nervous system, spontaneous recovery is extremely rare. So there's no known cure for spinal cord injury. And although spinal cord injury was acknowledged in the medical literature all the way back to the times in ancient Egypt, it's only been in the last maybe 30 years that we've been gaining some clinical ground and ability to be able to actually treat spinal cord injury in hopes of restoring lost functions that include motor function, the ability to walk, and also sensation, the ability to feel. So spinal cord injury is tricky because injuries to the central nervous system pathophysiologically are very complex. So you have a cascade of a number of injury pathways that limit the ability of the CNS to spontaneously regenerate itself. So it's been observed since the early 1900s that central nervous system neurons rarely spontaneously regenerate. And this is due to a multifactorial injury process. And one of the primary inhibitory factors here is the establishment of a glial scar that you can see over in the schematic. And so this is the proliferation of a cell called an astrocyte in the central nervous system to wall off the site of injury and prevent the further spread. But this injury site is also very hostile. It's packed with immune cells. And so if you were to introduce an exogenous stem cell to try to replace the damaged tissue, often these cells die. So it's very inhibitory both to regeneration of host cells that are injured, but also new cells that are introduced. And so my project has actually been very fun and rewarding because it combines a lot of unique aspects in multiple fields. So it includes aspects of developmental biology, stem cell biology, neuroscience, and ways that we study neuroscience, like electrophysiology, biomaterials and hydrogels, device engineering, next generation sequencing and bioinformatics, glycobiology and the extracellular matrix, and also imaging platforms. And for this in vitro work, we are funded in part through SUNY Poly Seed Award funds. But then also there's this extension of the project, which is a bench to therapy development. So we're actually trying to develop something at the bench that can be used to restore function in spinal cord injury. And so in that part of the project, I get exposed to animal models of disease and also behavioral testing. And so altogether, this project is funded by the New York State Spinal Cord Injury Research Board. So we're trying to address some of these pathophysiological and technical needs in the field. And we're doing so in part by creating a multi-purpose platform that combines a number of modalities that are required to overcome these barriers to cell transplantation. And namely, that is stem cell technologies to provide the new cells that have the appropriate regional identity to integrate into the injury site, but also biomaterials that have multifunctions but are neuroprotective and allow the survival of these cells. In addition to other factors that convert the injury site from one that's hostile to one that is pro-regenerative. And so we're using stem cells to create neural stem cells, which can become any type of cell in the central nervous system, and also fully specialized motor neurons in the dish. And then we integrate these cells with biomaterial, so-called neural ribbons that you can see here in these cylinders, which are almost thinner than a human hair. And these are very robust. They allow the cells to maintain viability, and we can ship them overnight all the way to Houston, where they're then used as injectable modules into the injured central nervous system of rodents. And so ultimately the goal of this is to develop a multifunctional approach to overcome multiple barriers to therapeutic success in spinal cord injury. And so we're using hydrogel biomaterials, stem cell technologies, and also other approaches, just in hopes of restoring lost locomotor modalities that are lost after injury. And so this is a multi-institutional effort, and I would like to thank my advisor, Dr. Janet Palliu, as well as our collaborators, Dr. Phil Horner in Houston, Dr. Jose Chabeli in Michigan State, Dr. Robert Linhart at RPI, and Dr. Yubing Z at SUNY Poly, as well as Dr. Annalisa Samemi at UAlbany. And then thank you, I'll take questions at the end. And so Nushin Amini is the other PhD candidate in our lab, Dr. Palliu's lab, and she's not able to be here today, so I'll just show her picture and her slides briefly as well. So Nushin's talk is entitled Bioengineered Platforms for Human Stem Cell-Based Diagnostics and Therapeutic Interventions in the lab of Dr. Palliu. And Nushin is in the 5th year of her PhD and is scheduled to graduate this summer. And she has a history in biomedical engineering. So that's a picture of Nushin. Here she introduces the ability of stem cells to be used in regenerative medicine to treat disorders that stem from numerous organ systems. And she specifically focuses on diabetes and neurodegenerative disease. Here she discusses the different types of stem cells. There's multiple types, including embryonic, induced, and adult, that vary in their source. The different ways to analyze cells, and then also clinical relevance by complying with GMP manufacturing practices. So in this project, Nushin is sort of working with these matrices and modifying their surface so that they can be integrated with stem cell-derived pancreatic beta islet cells. And these are the cells in the body that produce insulin. And so these are perhaps the most clinically relevant cells to date because they've already been used in clinical trials and have been translated to different companies. And she's trying to use these different matrices with surface modifications to drive the maturation of these cells and the production of insulin in a bioreactor platform. And similar to my project, Nushin also uses stem cells to generate neural stem cells and other nervous system cells, such as astrocytes and the immune cells of the nervous system, microglia. And this is a device engineering project where she can actually study cell-cell communication on a chip and look at the way that neurons react with other types of cells in the central nervous system, both in a normal state and also in an injury state. And she's focused specifically on traumatic brain injury. And these are, this is the company that Nushin works with for the pancreatic project, bioreactor fabrication. and she also collaborates with the lab of Dr. Jun Wang at, I forget which SUNY institution he's at, but he's more downstate.
Okay, so I'm Chris Netspan. I'm a fourth year grad student working with Professor Dunn. I started with a chemistry degree in undergrad, and then I got a master's in chemistry doing particle synthesis. And now I'm working on chemical mechanical polishing slurries. So my research is focused on improving chemical and mechanical polishing slurries for CMP applications. The importance of CMP is that it creates the flat surfaces needed for semiconductor fabrication. On the left is a device made without CMP between depositions, and on the right is with CMP. And with those flatter surfaces, you have the capability of having more lines, easing the scaling to smaller devices. So CMP works by pressing a wafer against a rotating polishing pad that has a slurry being dropped onto it. The polishing pad shown on the bottom is porous, and the slurry has different chemicals doing the chemical part of CMP, and then abrasive particles for the mechanical part. A slurry can have as simple as water abrasives and pH adjusters, or additional chemicals like oxidizing agents and complexing agents. The slurries that I work on are serious slurries, using ceria as the abrasive, because ceria is both chemically and mechanically active for its polishing. And I'm working on trying to determine how to harness the chemical environment of ceria to improve these polishing aspects. We found that the 3+% of ceria is a key component for chemical removal. Ceria is redox active and switches between the 3 and 4+ state. And that ceria is it mimics the enzyme catalase through the reaction shown right here by consuming peroxide and switching from 4 to 3+, and then back to from 3+ to 4+. Now, large particles have high mechanical removal, but they also have really bad roughness. And the large particles have a low 3+%. So we just found that if we add a controlled amount of peroxide, we can increase this 3+%, maximizing chemical removal to possibly improve the roughness characteristics of the polishing. To test this, we polished silicon oxide with ceria, which is a common target for ceria slurries. And the material removal follows the trends that we see in the 3+%. And on top of that, the highest removal rate is much higher than a commercial slurry currently being used in industry. Moving on to roughness, the lower number means better roughness. And while the commercial slurry had little effect on the roughness of the wafer, using our improved ceria slurry, we decreased the roughness by a factor of 2. The improvements and changes we made to the ceria slurry also allow them to be used for metal polishing. And these are, new and novel applications. And this one we were polishing ruthenium. We found that our ceria slurry polishes at 3 times the rate of a traditional ruthenium polishing slurry, and our roughness improved this time by a factor of about 4 over the traditional ones made with silica. So by understanding the chemical environment of ceria slurries, we can improve their removal characteristics. We've shown this through comparing it to a commercial slurry with silicon oxide. We've also shown promise for other polishing applications like copper, tungsten, and ruthenium. So I'd like to thank group members, metrology department, and SUMCO for funding my research.
My name is Maximilian Lear, and I'm a second-year PhD student working under Nate Cady, and most of the research that I've been doing has been electrical characterization tests on resistive memory, in my case, ReRAM. So I'm going to be talking about the research here at SUNY Poly that I'm doing. So data usage within processes has been increasing exponentially, and it's looking forward for that trend in the future as well. And one of the main factors that is increasing exponentially is sensor data, which is largely a very short-term data component. And the idea, for an example, if you have a self-driving car trying to recognize a stop sign, you want to accurately figure out that it is a stop sign, and you want to do it in a very, very quick manner. Otherwise, the data is useless. So the need for current and future architecture to handle this large data amount quickly becomes the essential issue. So one of the ways that we can work towards fixing this is by taking away the bottleneck in memory processing speed. And I'm going to do the same thing as everybody else, and in the middle of my presentation, get my laser out. So currently we, for most things, we have a processing unit and a memory in different locations. And so if you had the example with the stop sign, you'd have the input of a picture of a stop sign, and throughout at one point in the processing, you would have to go and collect information from the memory to be used in your processing. Now, because these are in separate locations, a large proportion amount of time needs to be taken to actually move between these two locations. So one of the main things that we can do is remove the separate memory component and incorporate local memory in our processing units. So what we're doing is the creation of ReRAM memristors, where you can think of them as a capacitor type of structure. It has a bottom and top electrode where we're using titanium nitride in our cases, with a metal oxide insulator level. Now, there's two main states that you want to look at for your ReRAM devices. One is a low resistive state where a hafnium bridge is created, where we take away the oxide, and now we have a metal bridge connecting the top and bottom electrode. Now this metal bridge allows you to have easy transport of electrons, and on the opposite side you have a high resistive state where the bridge is broken up again by the oxygen and oxygen atoms, making it more difficult for electrons to move back and forth, creating a higher resistive state. And to achieve this, we vary voltage back and forth between a positive and negative amount, and when you reach a certain amount, it switches between the states. Now, this is very good for most cases, and there's a lot of potential that you can see in this, but with the main one being that it can store synaptic weight. So this means that this device is non-volatile. If I give it a specific resistance that I want it to be, it will stay on that resistance for an extensively long period of time. So no matter how many times I call it, it will always give me back the same value. And another thing that's very powerful for these ReRAMs, it has a lower area and power requirement than traditional non-volatile elements that you might find elsewhere. The analog performance is also very key for our ReRAM devices. So before I was talking about the switching between set and reset, or high resistive state and low resistive state. Now that due to, or you can adjust parameters for your devices to change what you want for those. So you can have a set resistance that you want to get, and by adjusting parameters, you can reach that. So our fabrication and processing at the SUNY Poly Fab, we've actually created 300mm wafers using the current 65 nanometer CMOS memristor process, which is a 1 transistor combined with 1 memristor, which means that the memristor is now locally placed by all of the processing that you're doing. And with these wafers, we've done characterization, including the creation and storing of a memory image, in this case, AFRL, which is one of the funding partners, Air Force Research Lab. And I've done plenty of other different types of electrical characterization tests, but this is mainly the research that I've been doing. So in conclusion, overall, your ReRAM devices allow for that bypass of a separate location for memory, allowing for faster processing due to its local position. It also has a lower power consumption in a smaller area, which means you can fit a lot more of them in that local area, not taking as much space up that you would in other cases. And that is my presentation.
Hello, everyone. My name is Tristan Head. I'm also a student in Dr. Nate Cady's lab. I'm on the bio side of things, though. And so here is a overview of my work as my PhD project. And so as you can see, I have some device development up here at the top, and then afterwards I get into computational simulations, and then also some biological experimentation as well. And so my project is very broad in that sense, which I find is very fun. But to give you some more background on what I'm doing, the goal of my project is to study the tumor microenvironment, particularly due to metastasis. So when you get cancer, what happens is you develop an initial tumor, and cells in that tumor will divide and grow, and then eventually enough mutations will build up that these cells will start to seek out new places to live. And so they'll migrate away from their tumor to some sort of vessel, either a blood vessel or a lymphatic vessel. They'll enter it and then take advantage of that flow to circulate throughout your body in a process called dissemination, and then leave somewhere else and set up shop there. And so this process of metastasis is very significant because it's responsible for about 90% of cancer-related deaths. But this procedure where the cells are entering the blood vessels and then leaving it is not well understood. And to study this, you could take the typical biological route where you study things in a petri dish, but now you're putting cells in a plastic plate and you're trying to equate that to complex biology, which doesn't translate very well. And so on our side of things, we want to look at these processes occurring in vivo. And so we need a tool to study it in that way. And then in addition to that, once we have this device, we can also begin to look at the mechanics of the tumor as well and how that's affecting drug delivery. So here's the device that I've helped develop. This is the microfluidic intravital window. So you can see one here on a mouse. And what it basically is a cover slip that we've surgically implanted onto a mouse over a tumor. And with this, you can knock out the mouse, put them on a microscope, and look at that tumor in high resolution down here. And then here's a up-close image of the window itself. And this is really great because now you can look at your tumor while it's still in a live animal. And with the fab processes we have here, we can now introduce chemicals or drugs through the window and observe them happening in real time. So here's a video of fluid being injected through the window. And here's a video of, it'll play. Let's see if it'll play. Oh well. Okay. So after I've developed these windows, the next step that we get into is simulating how they work. And so to do that, we've been using the software called ComSol Multiphysics, which allows you to bring in a bunch of different physical models from fluid dynamics to solid mechanics. And with these, you can begin to look at how applying a flow rate to the device can affect how the diffusion of molecules occurs inside of a tissue. And so with this, we can begin to predict with what flow rates can we expect concentrations of drugs to reach certain depths within the tissue. And then by altering these parameters, we can also tune this behavior to exactly the profile that we need. And so to wrap up, in this project, we build windows in the fab that we can then put inside of living animals to study biological processes. We use computational prediction models to characterize how these devices work. And then we're also, now we're getting into diffusion studies and some basic biological studies to get the fundamental parameters to put into these models. And I'll leave you with this video of a mouse that exhibits all of its typical behaviors with a window in it. It'll feed, it'll stretch. I think at some part in the video he'll actually like start climbing around. And so the windows are very well tolerated by the mice. They doesn't really seem to cause them any pain once it's been put in. And with using these, we can get a lot of valuable data as well. Yeah, so here's the mouse climbing around. Doesn't seem to bother him all that much. And so I'd just like to thank everyone that's helped me with the project, Dr. Katie, my PI, and also my collaborators over at Albert Einstein College of Medicine, Drs. John Condilas and Dr. Entenberg, and then my funding sources at NIH and the RNA Institute at UAlbany.
Hi, everyone. I kind of have a dual system going here, so I'm talking on my phone, and I hope you can see my screen, and if you can't, please shout it out or send a message. My name is Emma Rocco. I'm in my 4th year of PhD study under the advisement of Dr. Shahida Sandvik, and my thesis work is on the impact of surface polarity on quantum efficiency of cesium-free 3-nitride photocathodes, and this is a project that's in collaboration and funded by NASA and Jet Propulsion Laboratory. Photocathode detectors are a type of photodetector which utilize the photoemission process. And they capture incident photons, so this could be light from the sun or other stars and planets, and that light excites an electron within the semiconductor, which is then transported to the surface of the semiconductor, and if there's sufficient energy, it can be emitted and collected by an electrode over the surface. These photocathode detectors have applications in astronomy for mapping the intergalactic medium and determining the composition of planets and stars, as well as military applications such as missile detection and non-line of sight communications. And they're widely employed in image intensifiers, as can be shown here on the bottom left. The 3-nitride material system is particularly well suited for these UV photocathode detectors, as it has a wide and tunable band gap that lies within the UV spectrum. Additionally, it's chemically inert and radiation hard, which means that it will have a long lifetime and high reliability. And additionally, the 3-nitride material system inherently has a spontaneous polarization charge due to the crystal wurtzite structure. So this means that we can grow our materials in the gallium polar orientation, in the nitrogen polar orientation, and semipolar orientations in between, all having different spontaneous polarization charges. And this is important for us because in our photocathode structures, we found that in the nitrogen polar orientation, we have a lower energy barrier for the electrons to escape into vacuum. So using this N-polar material system can help us to improve the efficiency of our photocathodes. In all of our photocathode structures, we need a p-type absorbing layer as our bulk layer. Magnesium is the most common p-type dopant for the 3-nitride material system, but it has a lot of challenges, like self-compensation of the magnesium by the formation of nitrogen vacancies at high magnesium concentrations as a way to alleviate stress in the film, as well as segregation of magnesium atoms at high concentrations, which we can see through atom probe tomography reconstructions. So when we have a lower amount of magnesium, the magnesium is well dispersed evenly throughout the film. However, at higher magnesium concentrations past the solid solubility limit of magnesium in GAN, you form these cluster defects, and the magnesium in those clusters are not electrically active. There's additional challenges using the nitrogen polar orientation of GAN, in that you commonly form these hexagonal Hillock structures, which have semipolar sidewalls. as well as there's an increased incorporation of unintentional donor impurities like oxygen. However, these helix structures provide us an opportunity, as studies have shown that there's an increased magnesium incorporation in the step edges and semipolar planes, which our helix structures have. So to test if we can use this opportunity of the helix structures having semipolar planes to our advantage, we grew 2 photocathode structures, one on a low helic density nitrogen polar film and one on a high helic density nitrogen polar film. And we found that the photocathode that was grown on the high helic density film had a much higher quantum efficiency in this plot of quantum efficiency to the 1/3 versus incident photon energy, compared to the photocathode that was grown on low helic density, which only achieved 6%. And to confirm why it was that we have this increased quantum efficiency for the high helic density samples, we studied the magnesium distribution across a helix structure using atom probe tomography. So here we have reconstructions of S1, which is in like a planar region, flat region outside of this helix structure, moving into S3, which would be, well within this helic sidewall. And we see that the total magnesium concentration was roughly similar between all 3 samples, looking at these blue bar plots. But the cluster density of these magnesium clusters decreased as we moved into the helix structure. And this has showed us that there's an improved magnesium incorporation within the helix structures, which gives us a higher p-type conductivity of our films. And thus we have a higher photocathode quantum efficiency when we grow our structures on these high helic density films. And I'd like to thank the funding support from NASA and the Jet Propulsion Laboratory, as well as my advisor, my committee members, my former colleagues, and so many people at CNSC and other SUNY schools that made this work possible. I think that's one of the things that's so special about CNSC, is that everything is collaborative and we get to learn about each other's work and help each other so much. So thank you.
Hello, everyone. My name is Vincent Myers. I'm a 3rd-year PhD student in the lab of Dr. Shadi Shahedapur-Sandvik, the wideband gap optronics lab. And today I'll talk about a central part of my PhD thesis research, the microwave annealing activation of p-type GaN for future power electronics. Cue up my laser pointer. So first, I'll introduce the usefulness of GaN as a power electronic substrate. Power electronics in general is the conversion and control of electrical power through circuit elements. In solid state power electronics, some architectures that do this include FETs, field effect transistors, inverters, thyristors, IGBTs, other things that those with an electrical engineering background will probably be familiar with. Silicon has dominated this field since the 1960s, but we're reaching silicon's theoretical limits. So for future applications, very high profile, among which are automotive drivetrains, as is shown on the right in the kind of DOE schematic of all the different power electronics needed for future automotive applications, and other applications that have really strict requirements for power, weight, vibration, temperature. We need other substrates. So wide bandgap materials like silicon carbide and gallium nitride, as well as other materials within the 3-nitride system, are needed to develop these future devices. As you can see on the bottom left, the commonly cited Balaga's figure of merit that shows the theoretical limits of a material's on-state resistance versus its voltage breakdown, show the comparison of gallium nitride to silicon carbide and silicon. As you can see here with these measured data points, we can make silicon devices that pretty much meet the theoretical limits of silicon. A similar story is true with silicon carbide. Silicon carbide research is quite advanced and we can get quite close to the theoretical limits. However, GaN is still lagging a bit behind its theoretical maxima. For reasons that I'll explain. The high efficiency doping of GaN is needed in order to achieve the material system's physical limits. This typically means doping concentrations of both electrons and holes to in excess of 10 to the 19 per cubic centimeter. This is quite efficient for doping of N-type GaN. You can typically get activation of 90 to 95% of N-type dopants. However, in P-type dopants, typically only 2% of the P-type dopants, in the case of GaN, magnesium, are typically electrically activated. And getting much higher than this requires temperatures in excess of 1200 degrees C. However, temperatures this high often lead to severe degradation of the material. So we need more efficient P-type GaN doping to achieve these future high power solid state electronics. And to do this, we need to develop annealing technology to activate the material P-type without decomposing it. So I'll show you some of the approaches that other researchers in the field are taking to this problem, and then I'll transition to the focus of my research. So to dope GaN, one effective approach is through implantation, as I've shown in the schematics here. This allows control of the concentration of dopants as well as the area, so that the sometimes complicated structures needed for power electronics formation can be created. To activate these P-type dopants without using extremely high overpressures, it's required to anneal in a non-equilibrium regime. So because magnesium atoms can be electrically activated at timescales much faster than decomposition of the material takes place, GaN can be heated and cooled very quickly in order to take advantage of this. So these are three mechanisms that are commonly used to do this. One method is laser annealing, wherein above bandgap light selectively heats a surface in nanosecond regime pulses, typically to activate the magnesium. However, a drawback of this approach is that the active area is usually limited to areas of around a square centimeter. The most successful method to date is multi-cycle rapid thermal annealing, which is a non-equilibrium approach that allows annealing in these short, tens of seconds long, 1400 degrees C pulses to activate the magnesium with minimal material degradation. However, about 24 bar of nitrogen overpressure is required. Finally, the method that my work uses is microwave annealing, in our case by a gyrotron microwave source. It utilizes thermal and non-thermal microwave interaction with the GaN, and an advantage of it is that the beam couples selectively with defects and highly doped layers and can heat at very, very fast ramp rates of hundreds or thousands of degrees C per second and applied to large areas. So let's look at this in some more detail. A sample of a semiconductor, in this case GaN, is placed in a nitrogen pressure vessel with a microwave window at the top to allow transmission of the microwave beam. A gyrotron source operated at 60 gigahertz generates a microwave beam with a roughly Gaussian intensity profile that is larger than the sample, that allows the whole sample to be heated evenly. This is then read by a pyrometer. Similar to the SMRTA approach I showed in the previous slide, we can use this to rapidly heat the sample in recent experiments up to at least 1450 degrees C for just a few seconds at a time to activate magnesium without degrading the material. As you can see in the figures at the right and the bottom, microwaves absorb very differently in different materials depending on their properties. This absorption can be modeled as the attenuation of RF power in a lossy dielectric. The attenuation is exponentially related to decreasing resistivity, so the coupling becomes more efficient. It's also related exponentially to increasing field amplitude. So by increasing the power of the gyrotron beam, you can exponentially increase the amount of absorption. The interaction of the beam with the material also has non-thermal effects. Microwave resonance with individual defect centers in the material, as shown in the energetic schematic here on the right, can inject more energy into specific states than the bulk thermal energy of its surroundings. This produces a complicated set of effects, and therefore multiple approaches are needed to study it. We can study what's happening in the material by studying its luminescence, for example. Irradiating it with above bandgap light after annealing causes luminescence of defects within the bandgap that affect the electrical properties. Shown here, the GL2 and the UVL indicate or are suggestive of relative concentrations of nitrogen vacancies, native defects in the material, and UVL corresponds to activated magnesium. So a detailed study of this luminescence gives us information about the response of the substrate to an annealing condition. Also, by annealing a material with p dopants in one region and n-type dopants in another, we can form a diode. Analysis of the diode's performance gives us information on how efficiently the p-type dopants are activated. We can also study the response of the crystalline structure itself to implantation and annealing under various conditions with X-ray diffraction. Distortion and recovery of the crystal lattice has an effect on strain and the band structure of the material. Shown here, we can see the relative level of crystalline recovery after annealing under various nitrogen overpressures. Distortion and recovery of the lattice has an effect on the strain and the band structure of the material. So studying the effects of implant and annealing is critical to our ability to form devices. So in summary, by studying the electrical and optical and structural evolution of GaN together allows a deep physical understanding of the material to form, which in turn allows us to develop the technology to enable power electronics that future power electronics that I described at the beginning. I'd like to acknowledge the funding of the Department of Energy's ARPA-E for this work, as well as all the support staff that the previous students have mentioned. Thank you very much.
Thanks again so much to our students. You guys did a great job. And if there aren't any other questions, I'll just say goodbye for now.